Why did the viral vector go to therapy? It had a tough time dealing with all its host attachment issues. Why was the viral vector always the center of attention in the lab? Because it had a knack for delivering genes in the most viral way possible.
Oh.
There's got to be something in there.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Christina Bowens.
And I'm Jordan Ruggieri. And today we are thrilled to welcome another genius to the show, Dr. Clifford Froehlich.
Cliff is the head of a viral vector services lab in Plainville, Massachusetts, where he uses the Absolute Q and dPCR for a variety of amazing applications. Cliff's career in biochemistry stretches back nearly 15 years, and it's a pleasure to share more about his work, past and present, with you today.
Christina, you want to hear a viral vector scientist pun?
Absolutely.
Why did the viral vector scientist always bring a map to the lab?
Why?
Because navigating through gene delivery pathways is more confusing than a maze.
Amazing. Well, I'm super excited to have you here, Cliff. I've been looking forward to this all week. It's been my favorite conversation. I've been talking you up.
That's awesome. It's great to find a needle in a haystack.
So, Cliff, maybe we can hop right in. Can you give a little bit of background on yourself and what you do?
Yeah, so I'm Clifford Froehlich. I'm the senior manager and head of analytical development at the Viral Vector Services location in Plainville, Massachusetts. Our role there is to provide services to our clients to make various kinds of viruses for gene therapy. And so my role is to assist our bioprocess sciences group with providing the data that describes what they've been working so hard to do. And so without that data, it's just very expensive water. And so the data paints that landscape. It tells the story. And we utilize that to assess whether we were successful. And then also optimize things and improve things. Really, ultimately, in the end, the goal is to transfer all the intellectual property into the GMP side of the site and make drugs to help people.
And I know we're super focused here about digital PCR, but you see a lot of technologies in the work that you do. Can you talk about some of the different technologies that you see and encounter on a daily basis and the work that you do and kind of the work that the viral vector lab sees?
Yeah, I mean, so obviously, to start with, it's about essentially the 5 key attributes that need to be tested for any drug product that's going out. So, whether it's a safety test, whether it's identity, strength, purities, impurities, all of those things. And then obviously potency, efficiencies are all key. And within that, obviously the PCR realm, whether you're talking about Q, DD, D, all kind of start to feed into strength, into identity. We can assess some impurities utilizing that technology. But also where we come from is understanding empties versus full viruses. So not all viruses are immediately stocked with the genome. They can often be empty. There is mixed philosophy in the field of whether those empties are important or not. When we are dosing a patient, whether it's to clear the immune system so that the fulls can actually make it to the target tissue. So as we're going through, we want to understand, what's the quality? And when I say quality, it's a big Q quality. How good is it? For me, because it is going into a patient, because it could be my mother, it could be my brother, my sister. It must be the highest standard. And so the more that we know about it, the better. And we really don't want to limit ourselves to the technology.
Cliff, could you actually even go a step back and talk about what is a viral vector lab and how do you what's actually the ultimate goal that you're trying to achieve?
Maybe we need to take a step back even further and just look at the word. Obviously, viral is going to be a virus. So typically we are producing, manufacturing an AAV, which is more of our bread and butter at the Plainville site. That's going to be an adeno-associated virus. There are adenoviruses. So we all may be familiar with the J&J vaccine for COVID. That was an adenovirus. Then you could be looking at a lentivirus, or we could be even looking at a herpes simplex virus. All of these are non-replicating. They are referred to as suicide vectors because they cannot make a copy of themselves on their own. And they're glorified FedEx trucks to deliver a genome. And so that delivery truck in the science world is known as a vector. For these viruses that we produce, they're either carrying a single-stranded DNA, which is AAV, double-stranded if it's adenovirus, an RNA payload if it's a lentivirus, but all of them are coding something in order to target a gene that is maybe not correct. Maybe it got mutated through environmental reasons. Maybe it just got, transferred through homologous recombination from parents, etc. And for whatever reason, it's not functioning properly. And our goal is to be able to use this FedEx truck and put the correct gene sequence in place so that we can improve lives. There's a myriad of ways to apply this. That's where we are, utilizing these viruses as vectors to carry these genomic sequences to a person.
I love that. And I think that's that's honestly the reason that drives me every day to, do what I do. And that what drove me to continue following cool technologies and cool tools was the ability to, someday something that I do is going to impact real people. So I guess that kind of brings me to my next question is when we're looking at these FedEx vehicles, bringing these cool deliveries to really unique destinations, what about digital PCR makes it really useful right here, right now to help you in the work that you're doing?
I've done this for, I'm going to date myself, probably a little over 2 decades now. And I actually remember being in undergrad when PCR was invented. And to have seen the evolution of it is absolutely phenomenal. Going from standard curves of the qPCR to now absolute counts, absolute measurement, absolute determination, is extremely tight. And actually coming from a quality background where percent CVs, where you're measuring variance, and you have to mathematically fit to a logarithmic data set on a qPCR, it immediately takes your percent CVs that might be low at 2 or 4% and bumps it to at least one magnitude more, 20 or 40%. I think the other bonus for the Absolute Q is the addition of a robotic arm in the play hotels. When you're in my realm where I've got hundreds or thousands of samples coming in because we've got all these clients, we're screening all these conditions, et cetera, we can just go crazy with a robot and set these up and have them sitting in a hotel and let them run overnight. And so what it does it improves the ergonomics. It improves even the pipetting accuracy on that. And then we're actually utilizing the members of the team for what we actually pay them for, which is their brain power, not because they're a human robot. We instead have them go ahead and get the data that we need to accelerate our clients' roles, to accelerate their intellectual property, to get the drug to the patient.
Continuing on this digital PCR train of thought, when you are evaluating a new piece of technology, let's say it's the Absolute Q or really any type of new technology, what are your expectations? What do you look for? And how do you actually get that new piece of technology into your lab?
I think it takes multiple approaches. I'm a little bit more systematic, and so usually I kind of want almost a URS. What are my user specific requirements? For me, I have to keep in mind that whatever I'm going to develop or build in my lab needs to translate into a GMP ready lab, and so then it needs to meet all the validation criteria, the IOQs, the whole shebang, as well as the qualification of the methods. Then I usually have a demo Obviously it's first and foremost maybe a lunch and learn or some seminar. But then if we can actually be able to have a loaner in the lab for, a couple weeks to a month. But it really, honestly, Jordan, it really varies across the board. In all cases, I bring my team in because I want a diverse input. I need a diverse understanding of what people's opinions are, and then I can build a business case. And that's my role then, is to clear the roads for that, get the funding for that, and then we move forward.
Diving into even more on the why you're looking into this new technology. So I mean, look, you're in a regulated environment, right? You're working with customers that are trying to develop things that will end up in a subject or a person, right? How do you identify that you need a new technology? I'm assuming things aren't just going around and blowing up in the lab, right? So how do you actually sit there and say, I actually need something new?
Obviously we have to be up to date in the science, in the journal articles, et cetera. The FDA could demand it. So that's the second part. But then on top of that, as part of something that I require of my team, and I think it's best practice, is we trend all of our data. At least within my shop, we're going to have our own site reference standards running through most of our assays all the way through, no matter which client we are testing for, so that I can track and trace how well the assay is performing. And it's, that way it's not a client specific performance question. It's across the board. Is it an analyst, or is it the instrument? And is, let's say I've purchased an ELISA kit that's commercial, and I'm just always seeing it, where it bounces around by 25%. Well, that's a lot. That's an obscene amount. In some cases, I've seen ELISA kits where they may have an invalid rate of 25, 30%. By letting the data drive it, listening, going to conferences, staying educated, and listening to even the regulatory group who, FDA, the EU, listen, talk, collaborate, and assess, all right, is this good enough or not?
Awesome. I had a question because I think I heard kind of two different themes going. There's the first, which is a very like when we think about gene therapy and all the variety of diseases that it can really, all the variety of disease research that it can impact. It feels very custom, right? There's lots of different things that it can impact. And when we think about DPCR, we all have to think about novel biomarkers that we can track. But then I heard you also talk about some of the different quality and safety aspects that, you're using to look at in your lab. So what types of markers are you most often looking for? Are they the more standard, like res-DNA, viral titer quantitation markers? Or are you looking at more of these custom novel markers where you need, like, you don't want to be creating these novel standards all the time? What are you seeing more commonly come through your lab?
When we're talking about the gene of interest itself that needs to be, should be in the virus, usually we will make something special for that gene of interest. A lot of times those genes have been engineered. Some of them, so like, for instance, again, AAV, usually the capsid can typically only hold a virus or a viral genome no bigger than 4.7 kilobases. That's its limit. And so some genes are bigger than that, and so they're often engineered. In other cases, maybe it's a smaller gene, but they tack on different promoters, different introns, other things like that. And so what we usually do, and it's kind of twofold. We do need to make it specific to the gene of interest because we need to demonstrate, per the FDA, specificity. So if I've got two clients that are producing factor VIII, I need to be able to tell the difference between those two in the facility at the same time. But at the same time, I also need to be able to create amplicons of the genome across a unique portion of the genome or of the viral genome that is not inherently found in you or me. And the reason for that is one of our quasi-functional assays, quasi-potency assays, is a tissue culture infectious dose 50, TCID 50. All right. So here we take the virus that's been purified, and we're just checking to make sure it's infectious and what that titer, that infectious titer might look like. So we'll add it to HeLa cells. We'll infect it, we'll culture them, we'll grow them up, we'll go ahead and harvest it. And then what we do is look for that material, but we need to be able to discriminate that from the HeLa genomic endogenous genome. And so if I can make a span that goes from the gene of interest over into an SV40 domain or into the ITRs or into, the promoter that I mentioned before, then that's going to be unique. And so what that does is it works twofold, one for strength, the other for the attribute of more functionality or potency. But then Christina, to your, I guess the other dimension, which would be more the impurities like the host cell DNA, we could, at least in early phase, we can probably get away. And when I say early phase, that may be phase 1, phase 2 of a clinical trial. We probably get away with, just generally looking for ALU repeats, and these are repeats that are found in human cells and human genes. And what it does is it helps us quantitate, all right, how much of the HEK293 genome co-purified with the virus with it, that's an impurity, how much is still there? But I don't know what's in that basket. I only know that it weighs 20 grams. And so you could go general, and that's good enough for phase 1 and 2. Now, the caveat is that we're also expected to have very short contaminating DNA segments, no greater than 200 bases. And so a lot of times we'll utilize like capillary electrophoresis and things like that to understand whether we've got, do we have a full length gene? Or is it just a truncated part, which doesn't really matter? That's where our concern would be, is that a trouble? So there's a lot of factors and a lot of ways that this all impacts it. And utilizing, a dPCR approach like Absolute Q can assess the plasmid. It can assess, the residuals, the endogenous genome from the HK293s. And anything else that we may be concerned about as before we ever inject that product into a person.
I love hearing you talk about all the synergies, really, and how thorough the entire process is. I think that's what I really love the most about this conversation, because I think so much we hear and we think about, how is this one technology used to, to tell this story, to put this puzzle together. And really, it is truly a synergy of all these different tools in a toolbox coming together. Because this story has to be tight. This ship is run extremely thorough, and nothing is released without us being extremely sure that, that these products are ready to go. And I think that is like the most incredible part of this entire story, is just, how many touch points and how many, extremely intelligent people get together to make sure that the right thing is ready at the right time. So I love this. Are you looking to precisely quantify targets with digital PCR without a standard curve, measure viral titer, or quantitate residual DNA? Are you looking to scale your dPCR workflow while maintaining a simple and efficient process? If so, Applied Biosystems QuantStudio Absolute Q Auto-Run dPCR Suite is for you.
The QuantStudio Absolute Q Auto-Run dPCR Suite is an automated, easy-to-use dPCR solution that helps improve throughput and utilization for absolute quantification of your samples. The system enables efficient utilization with multiple users able to load and schedule runs, along with greater productivity with automated plate loading and run scheduling with a single click.
It also enables consistency and multi-plate analysis, so you can analyze batch sizes that match your experimental needs together for improved reproducibility.
You can learn more at www.thermofisher.com/autorun. That's www.thermofisher.com/autorun. The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
And now back to our conversation.
Awesome. All right. So, Cliff, welcome to what we are calling the career corner of the podcast. Can you actually, just give us insight How did you even get into this viral vector space? So what was your educational background and maybe even some of your career pathway to get there?
I think it's probably pretty similar for many people, a very circuitous route. I've always been a dork. I've always loved science ever since I was a kid, asking for a chemistry set from my mom and growing little microcrystals and whatnot because, well, I like to blow stuff up and test things and cook. Cooking is everybody's chemistry at home. So I say that because my background is I got my bachelor's degree in both nutrition and food science from Texas A&M University. I dug nutrition and I chose nutrition because I realized that maybe the first medicine is what you eat. I knew I wanted more education, so I stayed at Texas A&M and I got a master's in nutrition doing microbiology in a poultry science department, and I raised 400 chickens. And what I was looking for was the rate limiting amino acid in the nutritional feed of growing chicks. Because in the poultry industry, if you can optimize the diet, you can have the biggest, fattest bird that's sold by pound in the shortest amount of time. But when I walked into nutrition, I wanted to work in a hospital and I wanted to be a clinician and whatever. And so I ended up doing that. I'm actually a registered dietitian. I went after my master's as passing my boards after doing my clinical rotations and worked at the University of Texas Medical Branch in Galveston, Texas as a registered dietitian. And I oversaw the burn unit there. Orthopedic surgery, neurosurgery were under me. Medical ICU was also under me. And then I periodically had to oversee the nutritional support for the Texas State prison system that was attached to the hospital. So working with those inmates who are at various levels of their lives. Some are pregnant. Some are at the end of their lives. They all have the same challenges we all do. They're just in a different home. I found that I was able to do my work in about half a day, and I found myself sitting in the library reading Nature and Science every day because I really missed that science. I really missed nerding out over all of that, despite all helping patients and working with people and treating kids with diabetes and all that jazz. I missed it. So after a year of working as a clinical dietitian, I was like, I need a PhD. I've got to go on for my PhD. I was accepted to a few places, but I ended up opting to go to Louisiana State University, specifically the medical center up in North Louisiana in Shreveport, Louisiana. I joined the Department of Biochemistry and Molecular Biology, and there I was under the tutelage of Eric First. Eric First was one of the first postdocs of Sir Alan First, who literally wrote the book on enzyme kinetics. He was knighted for writing this book. And so I feel like I learned enzyme kinetics classically from essentially the man, but I was doing both single turnover and multiple turnover kinetics using stop flow fluorescence spectroscopy, surface plasmon resonance. And we started to dabble with the X-ray crystallography because here we were busy understanding the function of an enzyme. How does it work? It's a machine, it's doing something, it's turning something over. In our case, it was tyrosyl tRNA synthetase. So it was busy taking tyrosine, hydrolyzing ATP and connecting it to tRNA so that you could create protein in your body, one amino acid at a time. And so we were trying to understand how that enzyme could work, whether we could switch the chirality and engineer our own proteins from L to D amino acids. And really just try to dig into what can we do? How can we break the system? So that's what we did. And I absolutely had a blast. I really wanted to expand more on the structural side. So I made a beeline as a postdoc to St. Jude Children's Research Hospital. I studied under Eric Ennemark, who is a preeminent X-ray crystallographer. We worked on DNA helicases because if I could understand how to stop DNA replication, then we can stop cancer. Because every cell that's dividing cancer, all it is dividing cells. They all have to have a genome, but if they don't have a genome, then they're not functional cells. So if we can stop that, then if we can stop the helicase, then we're good to go. So we were working on that, and so I was able to be the very first individual to purify and determine the structure of a eukaryotic DNA helicase with single-stranded DNA bound to it. And so I was able to determine this down to 1.19 upstream. I could see the hole in the benzene ring of the phenylalanine of the proteins because I had such tight resolution. I could rotate it. I could see how the DNA is all binding to it. And it was a seminal paper. It was published in eLife. It was one of the very first papers when eLife first came out. And so it was really fun. It was an absolute blast. But then I needed a real job. And St. Jude's got a GMP facility there on site. And so I found out about a position that was available. I applied for it and I got it within the analytical development and QC group. We made monoclonal therapeutic antibodies, monoclonal antibodies. We had cell therapies. We had lentiviruses. So I was one of 20 people on a team to cure 11 little boys of bubble boy disease. We had kids who were flying in on private jets because they had to be ultra clean. And 6 months, 9 months later, they were getting on commercial flights. They're outside. They're living a normal life. Again, this is why we do what we do, right? And so I had a great opportunity there. And St. Jude is also home of a couple of WHO directors. And so we were working in BSL-3 suites, working on viral genome stock, or viral cell stocks, to essentially anticipate maybe the next pandemic. But I moved on from there to other companies to grow. And so while I was at St. Jude, after about 6 months at the GMP facility, I was hired in as the manager of QC. Everybody seemed to trend to come to me and talk to me and ask questions and stuff. So I cut my teeth and learned to lead and to guide and to mentor there. And so since then I've been some form of leadership, director, senior director, now to Thermo Fisher Scientific VVS as senior manager of the analytical development here. So that's a long story, but there you are.
I don't even know where to go from there. That was just inspiring.
In all reality, I didn't do it alone. It took a lot of really cool and really smart people. And, and that's the thing is I get to hang out with some really smart people. And usually most of those individuals are so smart that when they think, their brainwaves kind of come and slap me in the forehead. But, it's being able to ask the questions, not be afraid to ask the questions. I've gotten to a point where nothing embarrasses me.
Is that the advice you'd give? Ask questions. Is that kind of the advice you give to anyone?
Yeah, my advice is just don't be afraid. Nothing scares me anymore. I'm always embarrassing myself. If I had children, they would be constantly embarrassed. But it's just asking the questions, and some of them may seem really obvious, but no one asked it.
So we ask everybody actually who comes on the podcast. It's a perfect segue, actually. Was there something in your mind that stands out as your most embarrassing moment in the lab? And then on the other side, something that you're the most proud of?
I think that any drug product that I get out, I'm the most proud of. The fact that I've done what I've done so far is pretty cool, but I want more. I'm greedy. I want to save more people. Embarrassing. There was that time I dropped a bottle of Timid and it smelled like rotten fish everywhere in the lab, and it was absolutely horrible. So, again, I'm dating myself. I poured my own STS page shelves and we had Timid there as a polymerizing. Anyway, there is that. Obviously being elbow deep doing necropsies and chickens while you've got pizza sitting on the side because we're on a farm and it wasn't a true laboratory, and we were doing that during our masters. And when you're doing X-ray crystallography, obviously you need to do this usually on site. All those things we were very blessed because we could remote into the synchrotron and control all the goniometers and all the robotics, and we could actually collect all of our crystal data, essentially from a closet in my pajamas at 3 a.m. But oftentimes it's better to go on site. And, if you go to a particle accelerator ring, like a synchrotron, they're very large, and they usually, ours was argon, and they've got tricycles everywhere. So like at 3 a.m, you might be, having races going around in circles, literally, because it's a synchrotron, but on a big, massive tricycle. We all are kids at heart, so we just have a blast.
I don't have any other questions. Christina, anything on your end?
No, that is it for me. I just wanted to thank you so much, Cliff. This has been fantastic. Such a fun session, and a really, really great time talking to you. It's been excellent.
Well, thank you. It's been an honor to have been asked to do this. It's a pleasure.
That was Dr. Cliff Froehlich, Head of Analytical Development at Thermo Fisher's Viral Vector Services Lab, located in Plainville, Massachusetts. Thank you so much for joining us for today's episode of Absolute Genius. It was produced by Sarah Briganti, Matt Ferris, and Matthew Stock. With more great science around the corner in future episodes, stay curious, and we'll see you next time. Jordan, I have a good joke for you.
Oh, I'm always down for a good joke.
Considering we just heard a lot about chickens. This is a very popular one in my house right now, because I have a 5-year-old and a 2-year-old. Why did the chicken cross the road?
Why did the chicken cross the road?
Because... It's a classic.